Cool. So my research on the Virgin River is really a two-part study. The first of which is a population study, which seeks to answer what are patterns of avian richness and abundance at sites that vary in amount of tamarisk and the amount of defoliation by the tamarisk leaf beetle. And the second is a diet study, which seeks to answer how does the diet of breeding birds within and among areas with different amounts of tamarisk and tamarisk leaf beetle and other associated tamarisk insects. And we're going to investigate these questions through point counts, insect sampling, and bird feces analysis. Our field methods for point counts involve 6 to 12 points per site, and observers conduct point counts for a total of 8 minutes per point. Our methods for insect sampling, we adopted the Tamarisk Coalition Protocol and the Colorado Department of Agricultural insect sweep protocol, and sweeps are conducted at every point after point count observations were completed. And then our methods for bird feces collection involve setting up mist nets in breeding territories, and we target netted for 4 target species, that being Lucy's warblers, Abert's towhees, yellow warbler, and Bewick's. So we're doing this study on the Virgin River because, as you all know, the Virgin River has a lot of tamarisk. And we've got 6 sites. The furthest north is Desert Springs, Arizona, Beaver Dam, Big Bend, Mesquite, Gold Butte, and then our furthest south site is Mormon Mesa, Nevada. So the Virgin River is important in that the tamarisk leaf beetle is relatively new to the system. As early as 2009, there was hardly any tamarisk leaf beetle within the Virgin River system. In 2010, the tamarisk leaf beetle started moving into the lower Virgin River Gorge, and we saw high tamarisk leaf beetle abundance in 2012, and then in 2013 we saw low tamarisk leaf beetle abundance. Now we have point count data for 2009 and 2010, and also in 2013. And that's really important because this data is going to give us insight into bird communities before the tamarisk leaf beetle was in the system and also after the tamarisk leaf beetle was in the system. And so the consequence of high tamarisk leaf beetle abundance, as I'm sure we're all aware of, is what normally would be a green, foliated tamarisk tree gets munched on by tons of tamarisk beetles, and what you're left with is defoliated habitat. And if that continues to happen, if the tamarisk continues to be defoliated by tamarisk leaf beetle, we ultimately have tamarisk mortality. And that has potential to affect birds in that what normally would be a very high density of vegetation, which would provide cover for birds, for nests, and from predators, is no longer there if the tamarisk beetle moves in. And so what you're left with is a very open canopy, which exposes birds to predators and exposes nests to predators. And that also opens up nest sites for higher solar insolation at nests. And that's illustrated by this photo here, which is a southwest willow flycatcher that's sitting on a nest in a tamarisk stand that's been totally defoliated, and the temperatures are so high that the bird is actually panting to cool itself down. And so if we look at our focal species, we chose these focal species because these birds are going to respond to the tamarisk leaf beetle differently. Now this is supposed to be a tamarisk tree. Keep in mind I'm a graduate student. I'm not a graphic designer. So yellow warbler is going to stay high in the canopy, and they're going to build cup nests also high in the canopy. They're going to forage around higher in the canopy. They're going to jump between branches, picking off insects as they go. Lucy's warblers, they're kind of a weird warbler in that they nest in cavities. So they're going to occupy a lower part of the canopy, but they're still going to forage higher in the canopy, similarly to yellow warbler. Bewick's wrens, they're also going to be a cavity nesting species, and so they're going to occupy sites lower in the canopy for nesting, and then they're going to forage around the ground and also on the trunks of the canopy. And then Abert's towhees are going to stay pretty low in the canopy, they're going to nest really low in the canopy, and they're going to forage on the ground. So if we have defoliation, as you can see here, it's no longer green. Species like yellow warblers are going to be exposed to higher temperatures. Their nests, since they're cup nests, are going to be exposed to higher temperatures due to higher solar insolation. And they're also going to lose that foraging substrate that they require. Lucy's warblers, although their nests aren't going to be affected, they're going to lose that foraging substrate. And then species like Abert's towhees, they're going to be less affected by losing these canopy because of their nesting and foraging strategies. And so in 2013, we put our field sites into categories based on habitat and the amount of defoliation of Tamarisk that we saw. And at Mormon Mesa, we characterized Mormon Mesa and Big Bend as a dead Tamarisk site because we saw that greater than 75% of the Tamarisk there was dead. We characterized our Gold Butte and Desert Springs site as a live Tamarisk site because it showed greater than 75% live Tamarisk. And we characterized Beaver Dam as a native site because at that site, native willows and cottonwoods were a dominant species type. And then at Mesquite, we categorized it as a 50%, roughly 50 to 50 native to Tamarisk. And that again was just based on the habitat type that we saw at these sites. And so based on what we know about the species and their life history strategies and what we know about the sites in 2013, we can make some hypotheses about how these birds are going to respond to the Tamarisk leaf beetle across the 3 years worth of data that we have. So we would expect that warbler abundance will decline at sites that experience Tamarisk mortality. And that's because at sites that experience Tamarisk mortality, they're losing their foraging substrate and they're also losing their nesting habitat. We would expect bird abundance will remain constant or increase across years at sites that did not experience Tamarisk mortality. And that's because at these sites, foraging substrates and nesting habitat is going to be less affected by the Tamarisk leaf beetle. We would expect warbler abundance will remain constant or increase across sites or across years at the native and Tamarisk mixed site. And this is again based on the fact that at these native and this native and Tamarisk mixed site, we're going to see that there's less of effect of the Tamarisk leaf beetle, and the canopy required for nesting and foraging for the warblers is going to remain at these sites. And finally, we would expect warbler abundance will remain constant or increase at years across years at the native site, and that's again because at the native site, warblers are going to have that necessary foraging substrate and nesting habitat. So we look at the data. This is preliminary data. We have another field season coming up. But if we look at average detections at our dead Tamarisk site, Mormon Mesa, across the 3 years, we do see a decline in yellow warblers shown here in yellow, and Lucy's warblers shown here in green. And then we see a consistent numbers from Abert's towhees and Bewick's wrens. And this is a pattern that we would expect because at these dead Tamarisk sites, we don't have the necessary nesting habitat for the yellow warbler, and we also don't have the necessary foraging substrate for both of these warbler species. If we look at our live Tam site, our hypothesis being bird abundances will remain constant or increase across years at sites that did not experience Tamarisk mortality. Again, this is average detections on the y-axis, year on the x-axis. We see that numbers are relatively consistent across years. Given the error bars, the increases that we see in Bewick's wrens and yellow warblers, and the decreases that we see in Lucy's warblers and Abert's towhees, I don't know if they are significant just given the error bars there. But again, this is a pattern that we would expect to see consistent numbers because the canopy across these 3 years would remain constant. So the necessary habitat and foraging substrates for all these species is available at this site. If we look at our native and mixed site, Mesquite, Nevada, again, average detections across 3 years, we see kind of this weird pattern with yellow warbler decreasing. I'm not quite sure why this pattern is, because at this site, there is still the necessary components, habitat components for the yellow warbler, and we also see a decline in numbers with Lucy's warbler. It's less significant given the error bars for Lucy's warblers. However, there is this decline here, and then we also see a decline in Abert's towhees, which is an interesting pattern as well. Again, these are preliminary results, but it's an interesting pattern nonetheless. And then an increase with Bewick's wrens. So our native and Tamarisk mixed site are showing kind of strange patterns that we wouldn't necessarily expect. And then finally, at Beaver Dam, which is our native site, if we look at average detections across 3 years, we start to see patterns that we would expect, especially with yellow warbler. Average detections increasing over the 3 years. Lucy's warbler showing consistency across 3 years. Bewick's wrens also showing consistency across 3 years. And Abert's towhees, given the error bars, showing consistency across 3 years. So from these preliminary conclusions, looking across these 3 years, we see differences in species abundances between years. So if we just look in 2013, this would be post-Beetle, we can make some new predictions just about our species. So we would expect Abert's towhees abundances will remain constant across sites. We would expect Bewick's wrens abundances will be consistent across sites. Lucy's warblers would be less abundant at dead Tamarisk sites, and yellow warblers would be more abundant at native sites. And just based on the life history strategies of the Abert's towhees, we would expect that average detections across our sites would remain consistent. However, at our Desert Spring sites, we see lower numbers, and given the error bar, that could prove to be significant given another year of data. At Bewick's wrens, we would also expect to be consistent across years, across sites here, average detections. But we're seeing in dead Tamarisk sites higher numbers. We're seeing in the native Tamarisk, native and Tamarisk site higher numbers, but in our live and native, we're seeing lower numbers. So that's an interesting pattern for Bewick's wrens. Lucy's warblers, we would expect to be less abundant at dead Tamarisk sites because they lose the foraging substrate, the necessary foraging substrate. However, at Big Bend, which is a dead Tamarisk site, we see higher numbers. And then at our live Tamarisk site, we see lower numbers. So that's an interesting pattern with Lucy's warblers. But we do see the highest numbers at Beaver Dam. And so another year's worth of field data will make sure that to see if these patterns are consistent. So yellow warbler, this is a pattern that we do expect. We see higher numbers in our native sites. So our preliminary conclusions for 2013 is that we do see differences in species abundances between sites. And so what could be driving these trends? It could possibly be diet. So I looked into this through a traditional approach, which is to take fecal samples, shown here, and under a dissecting scope, try to pick out identifiable insect parts. And if anyone's ever done this, they know that it's time-consuming. I hope you haven't done this, because it's extremely time-consuming. And it's also extremely difficult. As our friends from St. Francis showed us, they can't even identify whole insect parts. Imagine just fragments of insects from fecal samples. Other times, it's much easier. Like from this Lucy's warbler here. If you can make it out here, you see this really obvious gonzo nose that belongs to our friend Coneatus splendidulus. And then what about the Tamarisk leaf beetle? This is the only evidence I have so far of Tamarisk leaf beetle, but it appears to be the tarsi of an adult Tamarisk leaf beetle. So I've only shown you hard body parts, but we know that insects also have soft body parts, and larvae are primarily composed of soft body parts. So if insects are eating larvae, we could be missing all of the soft body parts that pass through a digestive system. So we're developing a new approach to increase our detectability of prey species in bird diets. And that's using the cytochrome oxidase 1, or the CO1 gene. This has been shown to be successful in analyzing bat diets, and we're trying to develop it in birds. And CO1 is a highly conserved gene, meaning that all insect species are going to share this gene, but within species, it's going to be different across species. So the approach is similar to the traditional approach, where you start with bird poop, and you extract the DNA, and then you make millions of copies of the CO1 gene using PCR, or polymerase chain reaction, which is just a process of heating up and cooling down the DNA. And by doing that, you create millions and millions of copies of DNA. And then with that DNA, you sequence the CO1 DNA, and that'll give you individual species sequences. And you'll be able to tell, based on the DNA that you find in your bird poop, if it what prey species it belonged to. And so will that work for us? Well, we've been successful in amplifying CO1 genes in reference insects and also in fecal samples. And this is an image of our electrophoresis gel after PCR. And electrophoresis is how we visualize DNA. So for our reference insects, it's pretty obvious. These bright bands here indicate proper amplification of the CO1 gene. So it's really obvious in our insect samples. In our fecal samples, it's not quite as obvious. But you do see a couple bright bands here, here, here. And so all in all, there was 7. And although for our fecal samples, the bands are faint, the fact that they're still there means that we were successful in amplifying that DNA. And this could really change the way that we look at bird diets. And that's because using this approach with the CO1 gene, we can look at more samples and process these samples faster than you could through the traditional approach. So if we're continue to be successful with implementing this approach in birds, this could totally change how we look at bird diets. So preliminary conclusions based on the first year of this study. We see differences in species abundances between years. We see differences in species abundances between 2013 sites. And we're successful in CO1 amplification in a subset of fecal samples in Tamarisk-associated insects. And so I've had a lot of help on this already just in the first year. I especially want to thank the MGen lab at NAU for helping me with the diet analysis portion of this. And with that, thank you.
I'm very impressed you actually could amplify DNA out of poop.
Thanks.
Was that 100 base pair ladder on your? So
Yep.
That's only a 200
Yes.
base pair front.
Yep. Yeah. So I didn't want to get too bogged down with the details, but Zainab is bringing up what these images are, what these bands are, and these are base pair ladders, and basically they're reference for determining how many base pairs your gene is. And so for the CO1 gene, it's about 200, and so that would be indicated by looking at the base pair ladder. Yeah.
Okay, that's a pretty small part of this code.
Yes. Yep.
Let's talk about it later.
Okay. Yep.
Okay.
Sure.
I was doing a project on monitoring fish populations. We had to look at their diet too. And we just electroshock them and cut them open, check that way. Simple. Would you have any thought of doing that? Not necessarily killing the birds, but if you find those that died peacefully of old age.
Well, yeah, when handling birds, generally the practice is to hold them for a little amount of time as possible. So when we were collecting the fecal samples, we only held the bird for about 10 or 15 minutes. And we would rarely find dead birds to analyze their diets. The other option would be to make them throw up, basically. But that's kind of stressful for the bird, and it's better to just collect their fecal sample after holding them for 10 minutes, than make them throw up a meal that might have been really hard for them to find in the first place. So yeah, our goal is to be as uninvasive as possible, but still collect data that would be really useful.
You mentioned that you had some higher detection levels with the dead tamarisk with some of the species that you weren't expecting. Could that be due to it just be easier to see the birds, whether there's more defoliation?
Possibly, yeah. I don't know if you've ever been to a dead tamarisk site like Mormon Mesa or Big Bend, but these sites are so dense that it's extremely hard to walk through. It's even really hard to see through. So I don't want to make any conclusions too soon, because this is only preliminary data, and so after another year of data, and we see similar patterns, then we might be able to make some conclusions. But as for right now, this data is unadjusted for detectability, and yeah, that's a good point, though. Thank you.
I don't know what most good ones, I was just wondering how